Hey guys, and welcome back to this channel, and this week's video is going to be biology themed, and it's going to be about glycolysis, which is a step in cellular respiration. And this video is focused on, again, the IB curriculum, but if you're from another curriculum, I bet you can also have a lot of information that overlaps with your syllabus, or just simply watch for the sake of curiosity, because I think this is pretty interesting, because this is how we all get our energy. And so let's start off right away. Glycolysis is the first step in both aerobic and anaerobic respiration. That's because glycolysis itself does not require oxygen, and it occurs in the cytoplasm, so it doesn't need any specialized organelle, like for example the mitochondria. We start off in glycolysis with a glucose molecule, and I'm going to draw 6 circles, so to say, because glucose has 6 carbon molecules. If you remember, glucose is C6H12O6, and so whenever I draw circles, I'm just representing the carbon molecule so that you can see what's happening. That I hope it will make better sense for you. So the first step in glycolysis is phosphorylation. Now, phosphorylation, like the name might suggest, is when we add a phosphate group to a molecule. And this phosphate can come either from ATP, right? Because ATP is adenosine triphosphate, so it has phosphate groups. Or it can come from, for example, free phosphate in the cytoplasm that is just flowing around. And basically what we do is we simply add phosphates to our glucose. Now, we end up with a molecule that has 6 carbons and 2 phosphate groups. So in order for this step to happen, we need to get 2 phosphate groups from somewhere, right? They can't just appear out of nowhere. And in this case, we're going to use 2 ATP molecules so that we end up with 2 ADP molecules and 2 phosphates. And these 2 phosphates are going to contribute to this molecule that we have right here. Now, you don't have to know the name of this molecule, just know that it has 6 carbons and 2 phosphate groups, and don't worry about it too much. The next step is lysis, which is simply the breaking apart of a molecule. Now, why this happens is because phosphate groups are quite negative and they create a lot of repulsion and make a molecule unstable. So I'm telling you all of these details so that these steps make more sense in your head and you can more easily remember them because they're more logical. So again, phosphate groups cause instability and the molecule lysis. So we're going to end up with 2 molecules that have 3 carbons each and 1 phosphate group each. Now this molecule, you do have to know how it's called, but you can know the shorter version. This molecule is called TP. I'm just going to expand it for you so you know what it is. It's actually triose phosphate, and that makes pretty good sense because you have 3 carbons and 1 phosphate. And our next step in glycolysis, and there's 4 steps total, so we're halfway through, is actually a little bit more complex because there are 2 things happening at once. The first part of this is oxidation. And you don't have to know an order. There is no really an order. These steps happen at the same time. We oxidize TP, and again, we also undergo phosphorylation. So let's start off with the oxidation step. Now, if you're not familiar with redox reaction or what oxidation or reduction is, I'll quickly explain it for you. Just remember the mnemonic oil rig, and it basically stands for that oxidation is loss of electrons or hydrogens, and reduction is gain of electrons or hydrogens. So in order for TP to be oxidized, it has to lose electrons. Now, electrons don't just disappear. They have to go somewhere because they can't just freely flow around. They're super unstable and super negatively charged, right? So we need something that's going to accept these electrons. And that's where NAD comes in. And remember NAD because it's going to be really important. NAD is an electron carrier, and basically it's just going to take on these electrons that TP has lost. And by doing so, again, remember, reduction is gain of electrons. It gets reduced itself. So we're going to need NAD to help us oxidize TP, and as a result, we're going to end up with reduced NAD. Now we have 2 TP molecules, right? So we're going to have to oxidize both of them, and so we're going to need 2 times NAD and we're going to end up with 2 times the reduced NAD. Now, let's move on to the second step, and that is phosphorylation. Now, in this step, phosphorylation, or the phosphates themselves, are not gotten from ATP, so we're not actually using even more ATP. These are just free phosphate groups, right? And while it might seem counterintuitive that in the first step we actually used energy to create energy, it all will make sense because in the end we're going to produce more energy than we make, and so it will be worth it for us, so to say. Now let's draw what we have ended up here after oxidation and phosphorylation. We again have 2 molecules, 3 carbons each, but then these molecules have another phosphate group each. So actually we're going to need 2 phosphate groups. And this molecule is called G3P, which stands for glycerate 3-phosphate. And again, you don't have to know the full name, but just be familiar with it in case it comes up. And now for our final step, this one is pretty easy to remember because this is the whole point of cellular respiration, and that is ATP formation. Now, like I've said before, ATP needs phosphates and has phosphates, right? So we have in our G3P molecule quite a bit of phosphates. In fact, we have 4 phosphates for our 2 G3P molecules. And so we're going to use and release these phosphates, and now we have free phosphates, and these phosphates are going to join together with ADP molecules to form ATP. So we have 4 phosphates, so we can combine 4 phosphates with 4 ADP molecules to form 4 ATP molecules. And this is a win for us because we have gotten more ATP produced than we used up. And as a result, we're going to have simply 2 molecules that have 3 carbons each, and these molecules are called pyruvates. So our initial reactant was glucose, and our final product was 2 pyruvates. And we have used up 2 ATP molecules, but we have produced 4 ATP molecules. So what we can say about glycolysis is there's a net 2 gain of ATP. Now this is not a lot, and in aerobic respiration, you actually go on to next steps which produce way more ATP, but in anaerobic respiration, this still counts, and so we still perform this step in order to get us at least some ATP. And so this is it. There's only 4 steps, and the more you repeat them, for example, just draw all of this out and speak through it or teach it to somebody, the more all these steps will click in your head and they will make sense and you will remember them. Because all of it is logical. I would say my key tips would be to know that adding a phosphate causes instability, because then the lysis step makes a lot of sense. And I would also remember that step 3 has 2 components to it, so you don't forget to include NAD or don't forget to include the phosphate groups. So I hope you enjoyed this video. I hope you found it useful. And let me know if you want to see a continuation of this, because I would make, for example, a part 2 with the link reaction, a part 3 with Krebs cycle, and a part 4 with the electron transport chain for aerobic respiration. But for now, just enjoy glycolysis and what it offers us, and see you next week.